So thank you, the organizer, for the opportunity. So we're interested in understanding how cytokine work, and this is really critical because this is the messenger how immune cells talk to each other. And many of the time, multiple cell types can respond to the same cytokine, yet have a very distinct function, and sometimes multiple cell types and this pleiotropic action. So the studies has been always how to decouple the cytokine pleiotropy in order to make cytokine more cell type specific. And this is important because most of the immune disease you can relate to the cytokines. Now, the last 20, 30 years, we've been studying cytokines in very optimal conditions. So in our lab, we have incubator, and we have very optimal conditions in terms of temperature, the pH, the oxygen levels, and nutrient-rich media. But very often, in the tissues, that's not the case, because the temperature can varies. Arthritis, you can have a very different temperature, in particular inflamed tissues. The pH can really drop to something around pH 5.5 to 6. And oxygen levels could drop from 20% to 1%. And also the nutrient could be different in different tissues. So for example, you can have much more potassium, sodium ions hanging in the tumor tissues. So I think this is really important to really study cytokine or cellular activity in the context of those chemical environment. So we are taking an approach of one cytokine and one environment at a time. So today I'm going to discuss about acidic pH, because this is a hallmark of the tumor because of the glycolytic activity of the tumor tissues. So we rationalize because immune cells, when they enter this pH environment, they really shut down the production of cytokines. And because the cytokine receptor interaction happens in the extracellular space, the chemistry of the extracellular space can really influence the ligand-receptor interaction. So we looked at varieties of cytokine, and we can say that at least 40 to 50% of the cytokine we have looked into has really impede activity under acidic extracellular environment. So we looked at IL-2, and we solved the crystal structure and looking into the acidic environment, how it can happen. We found that there is an histidine-arginine salt bridge in this critical interaction of IL-2's high affinity IL-2 alpha interaction. And as you know, the histidine has a tendency to get protonated. So when you have a H+ protonation, that cannot interact anymore with the arginine residue. So you have a repulsion. And that leads to this impede activity. Now, looking into the many other cytokines, we can really find the salt bridge interaction in multiple variety of cytokines. So in the lab, we use yeast display system to engineer proteins. So we rationalize if we can engineer a cytokine, or in this case IL-2, that could be more active in the acidic environment and less active in the systemic organs, we can perhaps switch its activity where it's needed. So just to give an example, an IL-2 is one of the first immunotherapy that we know of. And high-dose IL-2 therapy works, but it's only few patients respond. So we could show that either using a lactic acid deficient tumor, or in this case, sodium bicarbonate. If you use sodium bicarbonate, you could see that IL-2 therapy works much better. And this is direct effect, because we could look at the direct proximal signaling of this IL-2, and you could appreciate that in the tumor with a low pH, the IL-2 induced STAT5 is much lower. But in case of when you neutralize the tumor with the sodium bicarbonate, the STAT5 is really higher. So IL-2 therapy is highly sensitive to the acidic environment, and tumor unable to acidify their environment are really sensitive to the IL-2 therapy. So IL-2 variants that is highly thriving in acidic environment could have a really profound effect on the target while minimizing the toxic effect. So using yeast display, we created a molecule called Switch2. And Switch2 has this unique property that it has a very low activity in the systemic organs, bind to the receptor, very low affinity, but it have a full-blown activity in the acidic tumor. Shown here, the crystal structure of normal IL-2, and you can see the salt bridge interaction. Again, this histidine is there. It gets protonated. It has a repulsion. By our Switch2, what we've identified that we have an additional of different histidine, H35. So now this gets protonated and can interact with the aspartic acid. So there is this positive-negative interaction that Switch2 is enabling Switch2 to active in the acidic environment. So we think that this strategy could really be useful in terms of how we think about cellular activity and how we can modify cytokines in terms of making them more active in tumor environment. So we have done a lot of different assays to show that this is really the case in terms of looking at in vivo signaling, but just showing the tumor data and showing how IL-2 and switch could differ. So looking at MC38 model, we could show that we can at least cure 40% of the mice. And these are also inducing antigen-specific T cells. But one of the major effects of high-dose IL-2 therapy is toxicity. And by reducing the toxicity in the systemic organ and looking at here by edema and the lung weight, and looking into the pulmonary syndrome, you could see that they switch to have a minimal toxicity in the lung, which is very heavily oxygenated tissues. So just by changing how IL-2 interact in the extracellular space, depending on the pH, we could get this effect. I think our study also changed the 20 years of dogma of IL-2 therapy, that if you target IL-2 to CD8 cells or NK cells, as opposed to Treg cells, you could get better effect. And as you know, many of the trials, including some of the pharma companies here, have tried to make IL-2 more active to the CD8 and NK cells, and they have a huge toxic effect. I think what this study can show that, what we really don't realize that high affinity IL-2 receptor alpha is also expressed by the antigen-specific T cells, probably 10 times to 100 times more than the Treg cells. So we miss out when you're only targeting the dimeric alpha beta gamma receptors. So by switching the activity to the acidic environment, we could really target antigen-specific T cells, and thereby also, depending on the pH, can reduce their systemic toxicity. It also enables us to have a less sink effect and have a much more accumulation of the product inside the tumor, so thereby enabling us to have a higher concentration. So going forward, we are also looking into multiple different chemical factors in cytokines, and also hypoxia, redox as examples, and we're also looking to see how this cytokine can really boost the activity of the cellular therapy, because that's what even if you have CAR T cells, if you put in a hypoxic and acidic region, it does not make cytokine, doesn't kill the tumor. So we are hoping that by enabling CAR T cells to have this pH switchable, or we're also designing synthetic receptor of cytokine, we can make those much more effective in the hypoxic and tumor niches. And this is probably the most important slides. So this is led by Silvia Gaggero in the lab with multiple collaboration, and I thank you for your attention.